Hello, students. This next two video lectures are going to be a very short introduction to DNA. And so let's go ahead and get started. And I would stay focused on the PowerPoints because we're just going to be doing this at a very introductory level. So to understand DNA, we have to understand nucleic acids. And nucleic acids form deoxyribonucleic acid, which is DNA, and also ribonucleic acid, which is RNA. And the monomer that makes up DNA and RNA are called nucleotides. Nucleotides consist of three things: a monosaccharide, which is either ribose or deoxyribose, a nitrogenous base, and phosphoric acid. So those are the three things that compose a nucleotide. So let's look at the sugars. RNA contains ribose. Here's the structure of ribose. And DNA contains deoxyribose. Now what I'd like you to do is pause me for a moment and write these two structures down, and also with the numbering. Okay, so notice the numbering here: carbon 1, 2, 3, 4, 5. Carbon 1, 2, 3, 4, 5. Notice that carbon 2 is where the difference comes between deoxyribose and ribose. Ribose contains a hydroxyl group on carbon 2. Deoxyribose is just the hydrogen. The other really important part that I really would like you to highlight on your notes is the following. I'd like you to highlight this carbon here on carbon 3, and then this OH on carbon 5. Also important for us will be carbon 1 here. Okay, so these guys should be highlighted on both of your structures, and you'll see why in a few minutes, why I want you to know those guys and have a good feel for which atoms they are. Okay, so let's keep going. So nitrogenous bases. Adenine and guanine are nitrogenous bases that have as their parent molecule a purine. So it has the two-ring structure. Adenine is given by the symbol A, guanine by G. Okay, pyrimidines are a single ring structure. Cytosine is derived from that, so C. Uracil is U, and thymine is T. So the one thing to consider here is uracil is found in RNA, whereas thymine is found in DNA. And the other three are found in both. So A, G, and C will appear in both RNA and DNA, whereas RNA will have U and DNA will have T. Okay, first linkage up is a nucleoside. It's a compound that has the sugar and the nitrogenous base. So here, this is C, cytosine. Notice, looking at your notes, the linkage is between carbon 1 on, in this case, the ribose, attached to the nitrogen 1 on the cytosine. So you see how that's the linkage there? That would mean water came out when that formed. Here, this is the nitrogenous base guanine, and that's attached to carbon 1 on, in this case, deoxyribose. So always the nitrogenous base is attached to carbon 1. Why is it a beta linkage? It's because it's above the plane. Okay, all right. A nucleotide will have a phosphate group attached. So here, this is carbon 1, 2, 3, 4, 5. Here's our nitrogenous base again, attached to carbon 1. Here's the OH on carbon 2, so this is ribose. And then notice here on carbon 5, if you're looking at your notes where I asked you to write it down, carbon 5 has the CH2OH. That's attached now to a phosphate. Okay, this is actually the structure of ATP, kind of gives us a better feel for that structure. Notice that AMP is just one phosphate, ADP is two phosphates, and 3 is ATP. Okay, so this is once again going to show us a very important thing for the rest of our discussion. Here's our nitrogenous base, guanine. It's attached through that beta linkage, right, to carbon 1 on the ribose. See how that's ribose? Okay, now here is something very interesting, is that do you see how carbon 3 is attached to a phosphate? Okay, and then carbon 5 is attached to a phosphate that is also attached to the sugar. So what we're going to see here is that, in this case, this would be RNA. You have a backbone. Sugar, so this is 3, carbon 3, right? 1, 2, 3. Carbon 3, phosphate, right, to sugar. Phosphate, not shown, another sugar. So our backbone is going to be sugar, phosphate, and sticking out from that backbone will be the nitrogenous bases. If you can't see it on this picture, here it is again. Okay, here's that carbon 5. That, take a look at your notes. That's your carbon 5. And then it goes on that carbon number 5. So here, let me write it in here. 1, the carbon 1's always attached to the nitrogenous base. 2, 3, 4, 5. Okay, so carbon 1's always attached to the base. On carbon 2, it's just an H. So this is deoxyribose. Okay. Carbon 3 had the OH, but now it's attached to a phosphate on the other nucleotide. And so what you end up forming is the backbone is, look closely, sugar, phosphate, sugar, phosphate, sugar, phosphate. And then do you see how sticking out from that backbone are the bases? Not shown here. In DNA, this would be DNA because of the deoxyribose sugar, the base will be either A, G, C, and T for DNA. And then for DNA, that'll be A, G, C, or T. And then in RNA, do you see the difference here? Let me highlight it with a pen. RNA will have ribose there, but still the backbone. Sugar, phosphate, right? Sugar, phosphate. Okay, same pattern. The only difference is, again, the sugar is ribose, and then the bases will be A, C, G, or U. So the backbone is considered to be a phosphodiester backbone. Do you see why that terminology would apply? It's a phosphodiester, 2 esters here, right? Diester backbone. Okay. So again, phosphate, sugar, phosphate. So you see phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. You're getting the point. And then the bases stick out from there. Okay, now what this means is, the other thing is that this phosphate here was attached to the oxygen on carbon 5. So this is what's considered to be the 5' end. Over here, this is carbon 3 with its OH that could end it, or it could go attached to phosphate. So you have a 3' end. That's referencing carbon 3 and carbon 5 on that sugar. So if you take a look at your sugar structures, you can see that a little bit better. Okay, so a nucleic acid is a polymer with a free 5' phosphate group at one end and a free 3' OH group at the other end. That's on, again, referencing the carbons, right, on that sugar. The sequence is read from the 5' end to the 3' end. So in this case, we'd read the sequence A, C, G, T. Okay, now for DNA, the secondary structure is a very ordered arrangement of nucleic acid strands, and it forms what's called a double helix structure. In the double helix structure, they run anti-parallel to each other. So one of the strands runs 5', that would be this green one, to 3', and the other strand runs 3' to 5'. And in the middle, if you notice, it forms this very interesting arrangement of nitrogenous bases in the center. So here, one of the codes that's very important for us to understand is that they line up. A always goes with T, and C always goes with G in DNA. And what will hold this together is hydrogen bonding between the two nitrogenous bases, and also there'll be some hydrophobic interactions there from the fact that the base pairings stack on each other. You see how they're stacked like a staircase. So the base pairing is said to be complementary, and in this case, this is A and T. It has the ability to form hydrogen bonding between two groups here. So normally if you see them in pictures, it'll show these two interactions. The GC base pairings have 3 hydrogen bonding groups, and this does play a role when dealing with the idea that if you want to separate the two DNA strands, if you have DNA that's very rich in GC pairing, it's going to be a little bit more difficult to do because the interaction is stronger because there's 3 groups that are hydrogen bonding. Oh, wow. Okay, I don't know why all of a sudden it started making this noise. Okay, so this is just a recap of what I said. And so in DNA, 2 strands, they run in opposite directions. They're arranged in like a staircase. The base pairs are held together by hydrogen bonding. A goes with T, G goes with C in DNA. And each pair consists of a purine and a pyrimidine. And that keeps them the same width, right? The stacking of bases, you see how this is a space fill model. This stacking that goes on in the center is also stabilized by hydrophobic effect and van der Waals forces. More pictures. The other thing that happens if we look at the space fill model then is that DNA has a minor groove and a major groove. And that is going to be something that would play a role because there are some proteins that bind to DNA. We don't go too much into that, but that would play a role in why it's important that it has sort of this grooved nature to it. Okay, again, more pictures. This one's kind of cool because it's showing you a protein to DNA interaction. Okay, and we don't have time to really stress that part of it, but it is interesting. One type of protein helps the DNA coil up within the nucleus. So that's one thing. But other DNA could be involved in replication. I'm sorry, other proteins could be involved in terms of replication of DNA. So there will be some examples we see, but there's many, many different types of protein to DNA interactions that we don't have time to go through. And this is also just getting you to picture that the backbone, the center part where the base pairings are, they're stacking among each other, and that is stabilized through van der Waals forces. There's tertiary structure, which is there's some DNA that is circular, some that does supercoiling. So double helix can be considered to be like a coiled rope. And this coiling idea will be important because, your nucleus only has where the DNA is found in the cell, only has a certain amount of space. So you have to, the DNA has to somehow compress into a structure that'll fit in there. It does that by coiling. Involved in coiling eukaryotic DNA or histones, that's a protein that's very rich in basic amino acids. There's different types of them. Chromatin is a DNA molecule that's wound about a particle of the histones in like a bead-like structure. So this is chromatin. So chromatin then would be DNA plus histones. Okay, RNA is single-stranded, and it has a similar type of structure. Sorry, let me keep going back. And it's also going to have that sugar, phosphate. The difference is the sugar. It's going to be ribose. It's single-stranded. The base has this. It has the same structure, except you don't have thymine involved. It'd be uracil. I do think it's important here to note that it doesn't have that double helix structure, and so it has a lot more flexibility on the kinds of structures that it forms. So here's the statement on that it can form many unique folds and tertiary interactions. Okay, it can also form very complicated tertiary structures. So it's a lot more versatile in its overall structure than DNA. There are, this slide here it kind of creeps into it. It's probably one of the most important slides in all of biology, which is the central dogma of biology is that DNA can replicate, but DNA will code through a process called transcription to form messenger RNA. It's a type of RNA. And that mRNA is translated into protein. So as far as an introduction to this is a super important slide because this is kind of the overarching discussion that every biology major, every science major should understand, that DNA is transcribed into messenger RNA, and that's translated into protein. And for us, we did a whole bunch of discussions on proteins in this course. And so the DNA, the genetic code, codes for those proteins that we talked about, all of the enzymes, all of the transport proteins. And so if there's places where the amount of protein in the body can be regulated through from how much is made can be regulated on a DNA level. And that is something we don't have time to go into. So that's one important thing is proteins can be regulated through DNA as far as the amount that the cell it's going to have in it of that protein. The other part is that if there's any sort of mutation where the code is incorrect, that can lead to faulty protein, and that can be disastrous for an organism. So there are many diseases that are caused by an error in the translation process, the transcription and translation process that deals or the effect is to form a protein that's no longer viable for its function. Okay, so this slide then is really telling us how important the relationship is between DNA and the proteins that we've talked about in the course. Okay, so transcription, the sequence of bases in DNA is recorded as a sequence of complementary bases in mRNA. We're going to look more closely at that in the next video lecture. And then that mRNA is coded for specific amino acids that will be the primary structure of the protein. And what's going to help in that is another type of RNA called tRNA. And those tRNAs will interact with the mRNA and then bring the appropriate amino acid that's needed for that code. And then who's carrying this out are ribosomes. That's what you'd say is the cell's machinery for protein synthesis. So as far as what you need to know is really this discussion on this slide. I am going to go through in the next video lecture a little bit more detail with transcription and translation, but also understanding that if there's a mutation, if there's a change in this code on then it's possible that you have a protein that may not fold correctly, and then that can impede its function. Different kinds of RNA, transfer RNA, I would know that as the RNA that transports amino acid for protein synthesis. Ribosomal RNA, rRNA, is the forms ribosomes, which is where protein synthesis happens, and messenger RNA, which is the code, right? That's going to be the code that's transcribed from DNA that gives us which amino acid sequence we want. And those are the three that I need you to know off of this slide. Okay, messenger RNA, it's present in cells in a relatively small amounts. It's very unstable, has a fast turnover rate, single-stranded, and it's directed by the information encoded on DNA. And so we're going to look a little more closely at that in the next video lecture. rRNA makes up ribosomes, and ribosomes are the site of protein synthesis. Okay. And so I would know that as the definition of rRNA. tRNA, such an unusual and interesting structure. It is going to have a site that allows for it to bond to an amino acid, one tRNA per amino acid, and then on the other end of it, it has a site that will interact with the messenger RNA. Okay. So we're going to see that more in the next video lecture.